Hello students, I would like to welcome you back on the third and the last video lecture of the topic mutations. We are going to finish this topic, just the last topic is remaining, that is referred to as chromosomal aberrations. So welcome back. Let us just wind it up, let us just finish it up. In our last lecture we have already seen that what are genomatic mutations, how the genomatic mutations are present, which are the different types of genomatic mutations. Now what we have to study is about chromosomal aberrations. हमने अपने पिछले लेक्चर में देख लिया था जीनोमैटिक म्यूटेशंस क्या होते हैं, हमें क्रोमोसोमल एब्रेशंस देखने हैं. नाउ व्हाट डू यू मीन बाय क्रोमोसोमल एब्रेशंस? Chromosomal aberrations refers to the changes in the structure of the chromosome. ये क्या होते हैं? These refers to the changes within the structure of the chromosome. Changes within the structure of the chromosomes. Of the chromosomes is referred to as chromosomal aberration. इन्हें हम chromosomal aberrations कहते हैं. These can be of following four different types: deletion, duplication, inversion and translocation. What do you mean by deletion? In the case of deletion, a segment of the chromosome is being deleted. For example, you are looking at these two chromosomes. आप ये मान लीजिए ये दो क्रोमोसोम्स हैं आपके बॉडी के सेल के अंदर. इन दोनों क्रोमोसोम्स में ये क्रोमोसोम नंबर 1 की एक कॉपी, फर्स्ट की एक और कॉपी, अगर ये सेगमेंट डिलीट हो गया, ये सेगमेंट चला गया कहीं और, तो इसे हम क्या बोलेंगे? क्रोमोसोमल डिलीशन. अगर ये सेगमेंट डुप्लीकेट हो गया, let this be duplicated, then it is referred to as chromosomal duplication. Inversion, if the same segment has been inverted, then it is called as chromosomal inversion. And translocation, if the segment has been translocated from one chromosome over to the other chromosome and from the other chromosome over to the first chromosome, then this is referred to as chromosomal translocation. So these are the following four different types of chromosomal aberrations. Chromosomal aberration चार तरीके के हो गए. कौन कौन से हो गए? Deletion, duplication, inversion and translocation. अब हमें एक एक करके इन्हें स्टडी करना है और इसे वाइंड अप करना है. तो अब हम देखते हैं डिलीशन के अंदर क्या होता है. What happens in the case of deletion? Deletion refers to the loss of segment of chromosomal DNA is called as deletion. If there is a segment of chromosomal DNA that is being lost, then the process is referred to as deletion. Now you can find there are two different types of deletion. One is terminal and another one is interstitial. In the case of terminal deletion, what is going to happen? That from the terminus, the genes are going to be deleted. For example, there are 6 genes present over a chromosome. If one gene is being deleted, then it is called as terminal deletion. In the case of interstitial deletion, what is going to happen is that any segment of the gene is being deleted from anywhere, it is called as interstitial deletion. Interstitial deletion, any segment of the gene is being deleted from anywhere, it is called as interstitial deletion. Now when we are talking about terminal deletion, it is much more common as compared to interstitial deletions. An example of terminal deletion syndrome is Cry-Duchat syndrome. Cry-Duchat syndrome. This is the type of syndrome in which the cry of the child resembles with that of the cat. Let me just rub it. We will be focusing upon this syndrome, Cry-Duchat syndrome. The cry of the child resembles with that of the cat. The pitch of the voice varies from high to low. This syndrome is being caused due to deletion of a small arm of the 5th chromosome. This is being caused due to deletion of 5p chromosome. Chromosomal arm. 5th chromosome short arm. See, if there is a chromosome, then there are two arms. One is referred to as P, another one is referred to as Q. अगर एक क्रोमोसोम है तो उसकी दो आर्म्स होती हैं, एक को हम P कहते हैं, एक को हम Q कहते हैं. P refers to the short arm, and Q refers to the long arm. So, if there is a fifth chromosome present in two copies, and one of the arm is being lost, then this is referred to as Cry-do-chat syndrome. This condition will be that of Cry-do-chat syndrome. Okay, this is Cry-do-chat syndrome. An example of terminal deletion. We have already seen interstitial deletion, in which there was a deletion which was taking place from anywhere within the entire chromosome. Interstitial deletion. What was happening in the case of interstitial deletion? A chromosome was there. From any part of the chromosome, a deletion was taking place. Supposingly A, B gene is being lost, C, D and E is there. This deletion of B gene, is an example of here interstitial deletion. This is interstitial deletion. Okay, so first was terminal, second one was interstitial. We are going to divide the deletion into two parts. One would be the terminal, another one would be the interstitial. Terminal and interstitial. Terminal and interstitial. Okay. Now there is another thing, that is duplication. Now what happens in the case of duplication, that we must know. Now we are talking about duplication. In the case of duplication, the segment of the chromosome is being duplicated. Duplication के अंदर segment of the chromosome is being duplicated. Segment of chromosome is duplicated. Now duplication can be of two different types. One, it could be a random duplication, or another, it could be an inverse duplication. So there are two different types of duplication. The first type of duplication is tandem duplication. The second would be referred to as the reverse duplication. I must rub this out. I have to make some diagrams. मुझे कुछ diagrams आपको दिखाने हैं, इसलिए मैं ये मिटा दे रहा हूँ. Now what happens in the case of tandem duplication? Any segment of the DNA can be duplicated. DNA can be duplicated. Now how the segment of the DNA can be duplicated? For example, look here, this is a chromosome. There are genes which are present, 1, 2, 3, 4, 5, 6. Okay, now these chromosome genes undergoes duplication. For example, 1, 2, 3, 4, 5, 4, 5, 6. So these 4, 5 genes have been duplicated from here to here. This duplication is called as tandem duplication. इस duplication को हम tandem duplication कहते हैं, जिसके अंदर उसकी ओरिएंटेशन वही रहेगी, क्रोमोसोम की ओरिएंटेशन पे कोई फर्क नहीं पड़ता. Such type of duplication is found in the case of bar I of Drosophila. Okay, In the case of Drosophila melanogaster, what happens is that in the case of Drosophila melanogaster, the eye is actually divided into large number of facets. Okay? अगर आप ड्रोसोफिला मेलानोगास्टर मक्खी की आंखों में आंखें डाल के देखें तो आपको उसकी आंखों में डिवीज़न दिखेगा। वो जो डिवीज़न है उन्हें हम बोलते हैं फेसेट्स। मुझे पता है तुम लोग क्या सोच रहे हो, वो नहीं दिखेगा, डिवीज़न दिखेगा, डिवीज़न, एंड देयर्स डिवीजन्स आर रेफर टू एज फेसेट्स। ओके, किसी को मेरी मक्खी अच्छी लग रही हो टैटू बना सकता है इसका, ठीक है। दीज आर रेफर टू एज फेसेट्स। द जीन्स फॉर द फॉर्मेशन ऑफ दीज फेसेट is actually located on a 16A locus present on X chromosome. As there are two X chromosomes in the case of Drosophila, so I am making two chromosomes, 16A locus. This is also X chromosome. Okay. Now under normal condition there are 850 facets which are present. Normal condition में 850 facets present होते हैं. Under normal conditions, number of facets equal to 850. Now what happens? Tandem duplication occurs. Now what happens? Duplication. As duplication occurs, so this 16A copy is present in another form. This will cause the decrease in the number of facets. भाई जीन डुप्लीकेट हुआ है पर डिक्रीज हो सकता है, ऐसा नहीं है कि जीन डुप्लीकेट हो रहा है तो प्रोडक्ट हमेशा इंक्रीज होना चाहिए, डिक्रीज भी हो सकता है। इवन आफ्टर डुप्लीकेशन ऑफ द जेनेटिक मटेरियल द प्रोडक्ट कुड बी डिक्रीज्ड। सो इन such cases the number of facets in Drosophila gets reduced to 350. Hence प्रॉब्लम क्या आएगी इससे? प्रॉब्लम बहुत इंपॉर्टेंट है। The major problem that the Drosophila will feel because of the decreased number of facets is that it will not be able to visualize properly. The decreasing number of facets will inhibit its ability to visualize. And could be a prey to its predator. Okay, so this is what happens in the case of Drosophila melanogaster. After tandem, there is another type of duplication. The second type of duplication that is being found is reverse duplication. In the case of reverse duplication, the segment of the genes are being duplicated, but they are being integrated in a reversed manner. The segment of genes over the chromosomes are duplicated but present in inverse manner. In an inverse manner. That means if this is a chromosome, there are 1, 2, 3, 4, 5, 6, and this third and fourth get integrated, it would be like this: 1 2 3 4 4 3 5 6. See this, this has been inverted. This is referred to as reverse duplication. This is another type of duplication that we find in the case of chromosomal aberrations. Now the next is inversion. Now what do you mean by inversion? The process involves inversion of the chromosomal fragment. A fragment of the chromosome is being inverted, then the phenomena is called as inversion. देखिए, अगर एक क्रोमोसोम का फ्रेगमेंट इन्वर्ट हो जाता है तो उस फिनोमेना को हम इन्वर्जन कहते हैं. अभी हम समझेंगे कि इन्वर्जन कैसे होता है, क्या होता है. ना फॉर एग्जाम्पल, ये एक क्रोमोसोम है, मान लीजिए ये एक क्रोमोसोम है. अगर इसका फ्रेगमेंट टूट के यहाँ पर अटैच हो गया तो ये इन्वर्जन है. लेकिन इन्वर्जन भी अलग अलग तरीके के हो सकते हैं. One would be paracentric inversion, another one would be pericentric inversion. Okay, so we are talking about inversion. Inversion can be of two different types: paracentric and pericentric. Paracentric, the inversion which are away from centromere. कैसे याद करेंगे इसे? इसकी एक ट्रिक बनाएंगे. वो ट्रिक क्या है? You have to remember it by trick. The trick is paracentric. A means away from centromere. Okay. So paracentric inversion takes place when the inversion takes place away from the centromere. Such type of inversions occurs away from the centromere. You can look at the diagram. 1 2 3 4 5. There is an inversion. Inversion is taking place. Now it has been inverted. 1 2 4 3 5 away from the centromere, or you can write it like this: 3 5 4. This is inversion. This segment has been inverted. This segment of the chromosome has been inverted. This is called as paracentric inversion. Then there is another one which is called as pericentric inversion. Such type of inversion involves the centromere. Such type of inversions involves the centromere. For example, in the case of pericentric, it involves the centromere. This would be the trick. This one was the first trick. Now what you are going to find here is that this involves the centromere. So here the centromeric part will be inverted. First, second, third, fourth, fifth. This is the centromeric part. This part is going to undergo inversion. How? First, third, second, fourth, fifth. So this part has been inverted. This is the part that has undergone inversion. So this is referred to as pericentric inversion. Now the last one, the translocation. Translocation is a phenomena that involves transfer of chromosomal fragments from one chromosome to the other. Now how does this take place? Translocation. Just dropping this out. Last is translocation. it refers to transfer of chromosomal fragments. Transfer of chromosomal fragments. So there is a transfer of chromosomal fragments which is being involved. The phenomena is called as translocation. Now in the process of translocation, there are two different types of translocations that we are going to find. Which are the two different types of translocation that we will find? One is referred to as the reciprocal translocation. First, A type, reciprocal translocation. In the reciprocal type of translocation, the two chromosomes that are taking part in the case of translocation, for example, this is A, B, C, D, E, this is A dash, B dash, C dash, D dash and E dash. These two undergoes translocation. What we find is that in the first case, reciprocal translocation, if it is taking place like here, so there would be A, B, C, D dash, E dash, and here would be A dash, B dash, C dash, D and E. So here the translocation is taking place in such a manner that the part of the chromosomes have been reversibly exchanged. This part and the other part, this part have been exchanged. This part went here and this part remained here. Okay, this is referred to as reciprocal translocation. The second is non-reciprocal translocation. Non-reciprocal translocation. In the case of non-reciprocal translocation, what is going to happen? One part of the chromosome will move to the other chromosome, but no other part from this chromosome will enter into the other. For example, if this is A, B, C, D, E, this is A dash, B dash, C dash, D dash and E dash, which represent the alternate form of the genes, then what is going to happen? The DNA fragments will be transferred, A, B, C, D and E, and another one, for example, E dash is being added. There is another one, E dash, which has been added. And here it is A dash, B dash, C dash, D dash remaining, and a part E dash is being lost. This is referred to as non-reciprocal translocation. The last and the most important type of translocation, that is of utmost importance, is referred to as Robertsonian translocation. Robertsonian translocation. Question पूछे गए हैं इसमें से. Robertsonian translocation. What is this Robertsonian translocation? In the case of Robertsonian translocation, what is going to happen? In this particular case, there is a reciprocal translocation leading to the formation of a dicentric and an acentric chromosome. A specialized reciprocal translocation that leads to the formation of a dicentric and an acentric chromosome. Now, how does this translocation takes place? For example, there is a translocation that is occurring in between two chromosomes. There is the gene A, B, C, D, E. Okay, A, B dash, A dash, B dash, C dash, D dash and E dash. This is taking place. Now there is a cross that develops here. Now what happens? What happens is that there is a chromosomal fragment which is being produced from here till here, and this chromosomal fragment is again being produced. So here what you find is that it's something like a dicentric chromosome like this. Now the gene will be A, B, C, C dash, A, B, C, C dash, B dash and A, B dash and A dash. And another would be, along with this, there would be another one, DE, D, E dash, D. Okay, D will combine, E dash, D dash, D and E. The A-centric chromosome will be lost during the process of sexual reproduction, during the process of meiotic sac division. Note, very important point. The A-centric chromosome, this is A-centric chromosome. This is dicentric chromosome. The acentric chromosome will be lost during the process of gamete formation or sexual reproduction. Okay, so there is a loss of a very large fragment of chromosome, then such type of translocation is referred to as Robertsonian translocation. Hence, Robertsonian translocation leads to a loss of the major fragment of a chromosome, With this, we have finished the mutation in all of its form, the different types of mutations, whatever was being included in it, everything has been completed. Now we would be initiating class 12th from the first chapter, because this topic was very interesting, and that is why I thought, why not to take this topic. We'll be starting from the first chapter, from the first unit, and we'll be covering the entire syllabus. But from my side, this is all over. There would be a small lecture about the mutagenic agents, and that would be done in our next class. Okay, thank you, thanks a lot, and best of luck for your future. Okay, thanks a lot, students.